Thanks, Herbie. So just want to thank you guys that organized the conference for getting us all together. I very much look forward to it. I hope to learn a lot. I'm very new to extreme value statistics, something I think I only learned about a year ago from Ariel's book, actually. Simulator, the plugin. But what I want to tell you about today is the problem we've been working on for a while, which is trying to understand how cells build a filamentous structure of a very specific length. And just as a ways of introduction, I just want to let you know that this is sort of a problem that fits into a much broader set of questions that have been raised. Well, actually, going back to the 1800s, but here are some more recent works. And the question is, how do cells build up, assemble their organelles so that they match the size of the cell in which they're assembled? So, for example, these are some experiments that were done in Paul Nurse's lab. And in those experiments, they made fish and yeast cells of different size, doing all sorts of different things. Some were just wild-type cells. Some were wild-type cells, but diploid, haploid. Some had various genetic perturbations to them. And the result of all this was to produce cells of very different size. And here's just a simple plot of how the nuclear volume scales with the cell volume. And so that's the discovery that there's a roughly linear scaling. Similarly, one sees that kind of scaling. This is Wallace Marshall's work. When looking at mitochondria, this is again yeast, in this case budding yeast. There's a mitochondrial network, which is labeled in their experiments with some protein that's fluorescently labeled that's within the mitochondria. And they measure the total volume of this mitochondrial network, and they see, again, a very rough, very noisy scaling of the volume of this mitochondrial network with the cell volume, and some also experiments similar time from Tony Hyman's group on centrosomes, which are the sort of organelles that produce spindles. And this is in a developing embryo where you see as the embryo, the cells in the embryo get smaller, the centromeres get, their volumes also get smaller proportional to cell volume. So this is also true for spindles, for nucleoli. The thing about cells often is when you look at the cell and you look at the organelles inside the cell, the whole thing is sort of self-similar. Bigger cells have proportionally bigger organelles. And there's been kind of an effort in this community for many, many years to try to understand how does the cell achieve this scaling, what are the control mechanisms involved, and so forth. So what I'll focus on are filamentous structures. They raise sort of very specific problems of their own. Here are three of the sort of things that we've been working on. Flagella, and this is flagella of a parasite called a single-cell organism called Giardia. Those are the things that, at least in the US, I don't know how bad it is here in Europe, if you go hiking and you drink water, often the water will contain these bugs, and it's a bad deal if they get into your stomach. They won't kill you, but they'll make you kind of sick. They have flagella that are of very specific length. One would like to understand how is it that they program, how is it that they control that length. This is a very well-known example studied for many years. These are actin structures that come out of a single cell, which is a hair cell that's in the ear of every one of us. And you can see that, again, there's very well-controlled lengths of these things. There's also a graded structure. Some of the cilia are shorter, some are longer, and that actually is relevant for hearing. If you make a mutation where all these guys have the same length, the mice in which this is done go deaf. And finally, this is going to be the star of this presentation, a budding yeast cell in which actin filaments are again labeled. And there are these cables, which I'll talk about more. And we'd like to understand how is it the cell builds these cables. And as I'll show you, it builds them in a way that's just right. It's proportional to the size of the cell. And that's something that we'd like to understand. So that's sort of just the intro. Oh, and I was going to say, if you want to interrupt, please interrupt. I have some slides, but it might be much more fun to have a discussion. So if there's things about the biology that just sounds weird or confusing, please ask me questions. So Just for those of you who don't think about constructing a filament, I thought it might be useful to just get you in the right mind frame. So here's a little movie that shows if you were to take a bunch of actin proteins, so these are these little blobs, they're about 2.5, 3 nanometers in size from side to side. If you put them in a test tube, they'll diffuse around and they'll form filaments by attaching to each other. And if you do this in a test tube, in general, you'll find that the lengths of these filaments follow an exponential distribution. So if you plot the frequency of different lengths of these filaments occurring in solution in equilibrium. So this is not at all how these filaments assemble inside the cell. This is like a really bad thing for a cell if you just had a bunch of actin and it was just assembling into filaments all over the place. It's a bad design, and the cells do a lot of interesting things to avoid this from happening. And instead, this assembly is very regulated. It's regulated by a number of different proteins. I'll show you a really remarkable one here that's also figures in the story, which is called formin. This is the formin. And what it does is it nucleates one of these filaments, and then it actually assembles it. I think of this, for those of you who like Sesame Street, as the Cookie Monster, grabbing the actin monomers from solution and sticking them onto the growing end of the filament. So that's kind of the background that will be relevant for what I'll be talking about next. So there's my formin. So the structures that I'm concerned with are these cables. So this is a budding yeast cell. The bud is the place at which a new cell is formed. And for this new cell to form, you need, the way it's set up is the material that, the growth of this bud, is set up in a way where the material that's needed by the bud to grow is transported into the bud from the mother cell. So this is the mother cell via vesicles. These are these red things in this cartoon, which are transported along these cables. This is a very poor movie. The little things flying around their vesicles. I apologize, this isn't coming out very well. But here's a cartoon that's sort of depicting what you would have seen here. So the vesicles get onto these cables. They're covered with motor proteins, and they zip up. It takes a few seconds for them to get to the top. The dimensions here are a few microns, so they're going at microns per second, roughly. And the vesicles contain all sorts of proteins and other goodies required, including lipids, required to grow this bud. And if you make a genetic mutation that makes these cables too long, for instance, which we know how to do, the cells are sick. This transport is disrupted, and the bud doesn't grow as quickly, so there's a growth defect. So that just says, well, maybe we can't peer into the mind of a yeast cell, but maybe the yeast cell cares about the lengths of these filaments. So what we set out to do is measure the lengths of these filaments in yeast cells of different size. And that's something we wanted to understand, and that is how is the construction of these cables adjusting to the size of the cell. So similar to the experiments I showed you earlier in Paul Nurse's lab, we compared cables which are fluorescently labeled here. The little dots you see here are also actin structures, but they're patches. I'm not going to talk about them, even though they're also quite interesting and something we're actually working on these days. But the cables are these like long wispy structures you see in the mother cell primarily, and we can have cells of different size, either by looking at haploid cells, which means they have one complement of chromosomes, or diploid, 2 complements, they're slightly bigger. And then we can also do mutations. Here we have just a trick of a single mutation, whereby using temperature we can make this, we can arrest the cell cycle and allow the cells to grow very large. And here's the kinds of sizes of cells that we get in these different situations. Here's the sort of haploid, here's the diploid, and here's our mutant cells. And here you can see the size of the cell, which is measured as the the cell length. I'll be talking about cell length. That's just going to be the length from this bud neck to the south pole, the other side of the cell, denoted here with L. So you see that those lengths go between like 4 and 8 microns. So that's the sort of range that we've explored. And then we can measure cables, and what you can see immediately, two things. One is that the bigger cells have longer cables, and also, and this will be really important for my talk, you also see there's something going on with the variation in length that's getting also larger as the cell size gets larger. So I just want to point that right away out. So we'd like to understand this. The first thing that one of the first things we did is we said, well, let's look at the probability distribution of lengths, based on all this data. Each data point here is the individual cable that we've measured. And what I'm plotting here is the distribution of lengths, but scaled by the cell length. And what we saw is that these distributions seem to be lying on top of each other. So we seem to see, this is all seem in the sense that the data is limited, but given the best we can do experimentally, the distributions of lengths are scale invariant. So there's only one length in the problem, which is the cell length. It seems like the cell length, in other words, that's the whole distribution. An obvious or sort of a simple corollary of this is that if you plot the cable length versus the cell length, you should see linear scaling. And here we plotted a log-log plot, and the linear scaling is just the dotted line, and the full line would be the best fit. I come from a background in critical phenomena, and I'd be laughed off the stage if I showed this as any kind of evidence of scaling. But now I'm a cell biologist or a physicist or a theorist. I don't know what the hell I am, but this is the best we can do. So this is the puzzle. We'd like to understand how is it that you assemble this cable so that the distribution of lengths is scale invariant. Does that make sense? That's the thing that we saw and that we set out to understand. And there's really, and I want to sort of emphasize this, there's really two questions that these observations pose. One is the question of length control. I showed you an assembling filament, and I'll show you in a second movies of these cables assembling inside the cell. And so one question is, how do you arrange it so that as you assemble this filament, at some point you stop and you reach some fixed length? That's the question of length control. And I'll show you that the cell does that through a feedback mechanism. And the other question is, well, once you've achieved that length, how do you achieve scaling so that the bigger cells have bigger filaments? And this we've explored a lot, and I have quite a lot to say about this. This I have less to say about, meaning there's still many things we don't know about. In particular, what we don't know, I'll tell you right away, is we don't know the molecular mechanism of this scaling. We're still in search of that. But we have some good clues. So I'll be mostly focusing on this. So the thing that blew this whole problem open for us, we've been studying it for about 10 years in various ways, was the arrival of a postdoc in my group who's a cell biologist. He's now a professor running his own lab. But he figured out how to measure the dynamics of these filaments, these cables, in a living cell as a function of time. So he finds a cable where you can sort of follow some marker, which is there just by random chance, and you can see the cable extending in this cell. And what you'll notice is that it extends to a length of about 5 microns in about 30 seconds. It gives you a sense of scale. And the first thing we did was to ask, well, if we measure the length as a function of time, what we can figure out is the rate of extension as a function of time, because that gives us a sense of what might be going on. And the first surprise that we encountered was the following. So if you think about a structure that's like this filament that's growing towards the end of the cell, and that was what we assumed off the bat, is you might think, oh, the way it's going to know that it's hit the other side of the cell is if literally there's some kind of a mechanical force exerted on the cable as it sort of hits the other side. That would imply that the speed of the rate of growth should be roughly constant, and then maybe as it approaches the end of the cell, it should diminish. That's this yellow line, which we call the boundary sensing model. But in fact, that's not, that we don't see that. What we see is that as soon as this filament starts growing, so as soon as it starts leaving the bud here, it's growing from this bud neck out, as soon as it starts growing, it starts decelerating, meaning the rate at the extension slows down. So this is, we're interpreting this as telling us that there's some kind of a feedback where the rate of extension, as we see in this plot, the rate of extension decreases as the cable gets longer. Yeah, Herbie. That's a very good question. So, yeah. This is something we're also trying to figure out. So it actually gets to the end. It does a little bit of shimmy, which looks like maybe a treadmilling, and then it dissolves very quickly. So it undergoes something, and actually that's Shane's whole research, well, not whole, but part of his research program now. We have some theoretical reasons to believe, and he sees this experimentally, that they essentially do something like a dynamic instability. This whole cable, even though actin filaments themselves don't do this, so for those of you who are not experts, that's something that microtubules are known to do. It's a long story, it has to do a lot with their function, yada yada, but it looks like this cable effectively, and the theory actually predicts that, kind of grows, does this, and then has these catastrophic disassembly events. And that's what we see. It kind of grows out and then kind of very quickly disappears, and we don't have good measurements of the disappearing. So it might be treadmilling for a little bit. So then, okay, so this is some kind of feedback. Here I'm plotting the extension rate, the rate at which this filament is growing as a function of cable length. But now we look at two cells. These are our temperature-sensitive mutants, so small cells and big cells. And we asked, well, what does this feedback look like? And we saw that in bigger cells, that's the yellow guys, the slowing down is happening at a slower rate. There seems to be, the feedback itself depends on the size of the cell, to the extent that the yellow is different than green. And then if we replot this, where we scale the cable length, the length of this cable, with the length of the cell, then they seem to more or less lie on top of each other, at least within the errors that we have. So we decided to, yeah. They're going around the cortex. The one other idea was that it could be something about friction with the cortex that's slowing them down. Another idea, which we actually haven't explored to the extent that we should. We have few hundred cables, yeah. Somewhere in there I had, we can look at it later. Okay, so I like math and I'm a theorist, so we just decided to run with this observation and try to see what we can pull out of it. So what do I mean? Well, the fact that this feedback seems to be scale invariant. In other words, if we wanted to write down a rate equation for the growth of these cables, what we might start with is say, oh, the rate of change is some rate of assembly minus some rate of disassembly, and they could both depend on the length of the cable and the size of the cell. R, in this case, is the size of the cell. But what our experiments seem to be saying is that this function, this feedback function, is not a function of L and R separate, but a function of L over R. Of course, if this is true, then with a feedback function that looks like this, which would create a stable fixed point in this kind of dynamical systems description of the system, would lead to very simply to the scaling of the critical length or the steady state length with the radius of the cell, which is what we observe. So this, at least simple description, is embodies or captures some of these observations. But then we got greedy, or I got greedy, and I said, well, I've read my von Kampen book, and I can sort of turn this rate equation into a master equation. So if I have some rate of removing monomers and some rate of adding monomers with a stable fixed point where these two intersect, I can write down a master equation for the probability distribution because that's what I measure. And this is just what we write. There's nothing strange, there's nothing unusual about this. It just says the rate of change of the probability is there are two processes that contribute to the probability of a filament being length L, which is like, adding monomers if you're a filament of length -1, or removing monomers if you're a filament of L +1. And then, of course, they're both processes lead to the reduction of the probability, where if you start with a filament of length L and you remove or add a monomer, you will reduce the probability of being length L. And so if you write this down, you can actually do a lot with this. But one thing that I want to just remind you of, or tell you something, something I learned when I was just doing this problem a couple of years ago, is there's sort of this, again, you'll find it in von Kampen's book, there's this very general result that if you have a master equation that has this kind of form, in the sense that the length, in my case, at which the distribution is stable, or I should say the length at which the rates are equal, so this is the steady state length. If this length happens to be much greater than the size of the single unit, which in our case is definitely true because the lengths of these cables are 5 microns, the units are only a few nanometers, then there's a general result called the large systems, that comes from something called the large system size expansion, that tells you that the probability distribution of these lengths should be Gaussian, and the width of this Gaussian should be just a kind of a central limit theorem, should be just proportional to the square root of this critical length, the length at which the growth stops, times the size of the subunit. Okay. So that's a very general result, and it's independent of what the functional form of this K plus and K minus are. You can put in anything you like as long as you have some kind of a steady state where the two are equal. That's as depicted in this picture. So, this we did, and then for literally for 2 years we were puzzled because this completely contradicts our experiments. The first problem is these lengths are about 5 microns. This is 3 nanometers. If you put that in, you expect fluctuations of the order of about 100 nanometers. These are way bigger. They're like almost well over a magnitude bigger. The fluctuations are huge. And the other part is, if you look at how the variance of these fluctuations scales with the length of the cell based on this result, the prediction is that it should be the dashed line. And what we see is definitely not the dashed line. More something that looks more like the variance is proportional to the length squared, which of course again is directly a consequence of the thing that we also observed, which is that the probability distributions, when scaled by the length of the cell, all fall on top of each other. So this just also is another way of saying there's only one length scale in the problem. So there's no way a model like this could account for the fluctuations, no matter what these guys are. And that's how I got in. I feel like in one of those meetings. I've never been to one, I guess, Alcoholics Anonymous. This is how I got into extreme value statistics. This is when I hit the bottle. So I was forced into this. I did not want to get into this at all, but as it will, nature made me do it. The way we got into this is we kind of decided, wait a second, maybe we're doing something, reluctantly I say we did this because the last thing I wanted to do was actually start getting into the details of what the structure of the cable is. But what I didn't tell you so far, I've kept from you, is that these cables consist of many small filaments that are cross-linked. And here's an electron microscope image of one such actin cable. This happens to be in a fission yeast cell. No one's done it in my yeast cells, the budding yeast. But you can see that these are made of individual filaments that are about a half a micron in length. So if this half micron length scale is correct, then my cable would consist roughly, if you put them side to side, of 10 of these little filaments, right? Okay, so then how should we think about cable assembly and disassembly? And well, I told you there are these formants, and they're at the bud neck. So they're assembling the cables. They're all positioned here. There's a whole interesting story how that happens. So there are a bunch of these formants. Those are those guys that grab the monomers and stick to the ends, stick them to the ends of a growing filament. So here's my toy model. And what these formants are doing is they're injecting actin, and then there's cross-linking. I'm not drawing the cross-link, so what I've made is like a multi-lane model of a cable. So each one of these is a lane that's produced by one of these formants. You should imagine these things are all cross-linked together, so all these things are held apart. But now you have these individual filaments, and basically what's going on is there's mass going into the system from the boundary, but then each of these filaments can be disassembled by many other proteins. So then the filaments in the bulk are being disassembled. Now, if you just think about mass conservation, mass is going in through the boundary, it's leaving through the bulk, and if when those two match up, it will prefer a length scale. And so we think this is actually the feedback that's producing the finite length. But what is the length of the cable? What we measure is the extent of this whole structure. So the length of the cable is nothing but the length of the longest lane. So now with that in mind, and just saying, oh, there's some rate of adding rate of adding filaments. This is, we completely ignored monomers. The filaments themselves are some little L, which those EM pictures would suggest could be about 0.5 micron. There's some rate of removing these guys. Actually, we know all these three parameters from various other measurements, so there's actually nothing to fit, essentially. And then, then now all of you know how to do this. You can compute the cumulative distribution of what's the probability that the cable has a length L. For that to be true, it must be true that every one of these lanes has a length that's less than L. And so you get this product form, which is very typical of a Gumbel distribution. And in fact, if you sort this infinite product out, you do get a Gumbel distribution of the lengths, which is just a function of the parameters that go into the problem are just this parameter lambda. What is lambda? Lambda is literally the length to which a single filament gets to before it's dissolved. So these filaments are moving like on a treadmill here at a speed of L times K plus. That's the rate at which mass is coming in. And it'll get out, so that's the speed. And then if you multiply that with a time scale, which is the time over which this thing survives, that gives you the length scale lambda. And I should kind of wrap it up. So that gives you a Gumbel distribution. This Gumbel distribution then makes a very interesting prediction. So if we want to get this scale invariant distribution, what has to happen, and I won't go through the whole analysis, is that the length of these little filaments has to be proportional to the size of the cell. And that will give you a scale invariant distribution. This now has a really cool prediction, which we actually had to go out and measure. And that is that the number of filaments as you move down the cable should decrease exponentially. And the exponential rate of decrease should be proportional to the size of the cell. So these cables are getting dimmer and dimmer. The rate at which you get dimmer and dimmer is going to be slower in bigger cells than smaller cells. We measure that, we see that, and in fact, if we scale with the mother cell length, we see that this diminishes exactly as predicted in a scale invariant fashion. This kind of dynamics, where there's really no feedback through any molecular mechanism, but it's just due to the geometry of the cable, that really does produce this kind of feedback. For those of you who like doing calculations, you can kind of analyze this analytically and get the average length in terms of some exponential function. So there's, that black is the function, sorry, the dotted red line is this insane function. This is just some simulations which are easier doing this. So I'll just end by saying, well, this Gumbel distribution, as you all know, you can get almost regardless of what the dynamics of individual filaments are, as long as you pronounce that the length of your structure is the length of the longest filament. So in fact, if these filaments were made by a totally different process where the distribution of lengths was exponential, you would again get Gumbel. And what's interesting about Gumbel is that the variance, this should be L squared. The variance goes as L squared, and the ratio is basically a universal constant, except there's a log of n being the number of filaments, but in most applications, that's going to be just a constant. Just to entertain you, so my student, what he did is he found every measurement we can find in the literature of actin-based structures where people measured distributions of lengths. And these are all the examples we found. And we just took all of them, measured the variance, and plotted versus the length. And here's the scaling predicted by Gumbel. Here's the scaling you would predict based on rates that depend on L, the thing I dismissed for these filaments. It seems like, this is an interesting case, it seems like all these are different experiments done by different people, different decades. They all seem to kind of be indicative of this scaling that's predicted by Gumbel. In fact, the coefficient of variation comes out fairly accurately as well. So I'll just stop here and just tell you quickly what we've learned. Well, it seems like the cells know about extremal value statistics to the extent that if you want to build a cable of a length that's well defined, all you have to do is take a bunch of filaments, make them any way you want, and then just cross-link them. And that seems to be a strategy that's been used over and over again. So that gets us, and one of the things that predicts is that if you start doing genetic perturbations to this system, which we've done a lot, and that was 5 years of frustration, you will always see this feedback. And that's exactly what we saw. We saw the set point shifting left and right, but we could never get rid of the feedback. And so now we think we can't get rid of the feedback because it's built into the geometry of the cable itself. So that makes this whole thing kind of robust. And then, you know, I desperately want to think about evolution, and I can't help and I'm trying to think about how evolution might come up with this kind of scheme. Something that Gesper's been trying to do in the context of gene regulatory networks. And just finally, just to say, Shane is the great hero of this story. He's a cell biologist who bravely came to my lab to learn some mathematics and did some beautiful experiments. Now he does both, a bunch of grad students. Bruce is my good friend and cell biologist at Brandeis, who Shane worked with to get all these experiments done. And Predrag is kind of an insane math friend of mine from high school who helps me with all these things. And then there's funding. And thank you for your attention. And sorry, I think I might have gone overboard.